So I want to go over amino acids. It might seem a lot in the beginning, but if we break it down into its structure, it's actually fairly straightforward. So we do want to talk about all this different stuff. So building blocks of proteins, essential acids, their basic structure. They have a lot of side chemistry, which we're going to focus on today, and that's going to lead on to properties and ionization, which we will talk about later. So it's going to seem a lot at first, but when we break it down, hopefully into its parts, it's going to be very easily digestible kind of information. So first of all, amino acids are everything. So amino acids make proteins, and proteins are everything in our body. So sugars are energy, fat is structure and storage of energy, but proteins really do everything else. They're really structural support, but also all the little machines that kind of do everything. So they're the catalysts, they signal stuff, and they also create a lot of these mechanical actions within our cells. So they're really, really important. So we need to understand all the different components of amino acids, how they work, how they fold. And in order to do that, we need to understand the basic structure, which is amino acids. There's about 20 of them. There's 21, which is kind of like the rare one, but we're going to really focus on just the 20. So here's a list of essential versus non-essential. I think the name itself is pretty straightforward. Essential just means that you have to eat it. Your body cannot produce it on its own. Non-essential means that your body can make these on its own. So some of these you don't need to eat, and your body can still make it. Others you do actually need to consume. So here's a list of them. Do you have to memorize it? No, but I just like you to see the difference between the two. Now, before we get into amino acids, we want to talk about very, very common something called functional groups. And functional groups are really important because it tells you kind of what it does before you even look at the whole structure. So when we look at this, the most common one is ethanol, or an alcohol. So ethanol, remember, is 2. We'll eventually get to naming, but 2 is eth, 1 is meth, but in this case there's 2, so it's ethanol. This is the alcohol that's produced in when you drink alcohol, fermentation. Ethanol is the thing that's actually going to be formed. What's cool about this is that you have a relationship between oxygen and hydrogen. Remember back, I think like 2 days ago, we talked about electronegativity. So oxygen is very electronegative compared to hydrogen, so it's going to be a partial negative compared to its partial positive. Now, carbon is also going to be somebody who doesn't really need electrons. So all these electrons are going to be sucked towards the oxygen, which will give it a certain property. Because of its partial negative and partial positive, it can form hydrogen bonds. And because it can form hydrogen bond, that means it could dissolve in water. So even though this part right here on the left, hydrocarbon, it is a non-polar. That means by itself, it can't dissolve in water, but with the alcohol, now it can. So that's one cool thing that we have here. Another object that we have right here is a sulfhydride. So when you look at this, you're like, okay, well, it's a little bit different, but it's going to have very similar properties to oxygen. And if you look at the periodic table, you'll notice that they're right by each other. So because of that, you're going to have hydrogen that's also electronegative. And what's going to do, it's going to pull the electrons away from the hydrogen. So it's also going to be able to make hydrogen bonds. So those are very too common. They're very similar. You have oxygen and hydrogen and sulfur and oxygen. When you look at something like glucose and fructose, you'll notice, wow, there's a lot of hydroxyl groups. Oh, so this, these are alcohol, but you could also call them hydroxyl groups. And I'll kind of intertwine between them. So you'll see a lot of hydroxyl groups right here. But when you have in a linear one, you actually have this thing right here, which is a carboxyl group. And the carboxyl group is actually really cool too. You have a not very electronegative molecule in carbon. You have a very electronegative molecule in oxygen. So you get a partial negative and a partial positive. Later on, when we talk about chemical reactions, this is a source of we're going to have chemical reactions because the oxygen is very happy to steal back these electrons in this bond. You have a double bond, so technically there's 1, 2, 3, 4 electrons. Oxygen is happy to take these electrons back. Because of that, this carbon is very reactive, so it's very easy to interact with the other. But here, once again, you have a partial positive, partial negative, so that means it is hydrophilic. So all these things right now that I've talked about are hydrophilic because they can either make hydrogen bonds or they have partial negative, partial positives, which will kind of form the same thing that you have there. Your last group here is a phosphate group. Here is very similar. You have very electronegative oxygens that are going around. And when it's a relationship with the hydrogen, it's going to pull the electrons towards it. So you're going to have electrons that are going to go towards oxygen, make the hydrogens very, very positive. Once again, this can make hydrogen bonds. And it could also do something else. This can also be acidic. So a lot of these actually could be acidic as well. So this technically could be acidic. This one technically could be acidic as well. The carboxyl group for sure could be acidic. So I will put that right here, acidic. And the reason why I mean by acidic is because the oxygen so wants electrons, it can give away hydrogens in the right condition. And that'll make more sense as we talk more about it. But that's what acid is. Acid gives away protons and a base will accept it. So these are kind of the main ones that you'll see when we're talking about functional groups. So you have a hydroxyl group, which is an alcohol. You have a sulfide group, which is kind of similar to a hydroxyl group. You have a carboxyl group, which right here. And then you have other examples of oxygen and hydrogen. So here's another carboxyl group right here. All right, so here is an amino acid. Let me get rid of all this stuff. And you'll notice that you have a bunch of stuff. You have a carbon with a double bond here, a carbon with the OH, which makes your carboxyl group. You have right here another carbon with a hydrogen, but then you have something called amine group. So amine is also something that's very electronegative. So that means it's going to kind of, so nitrogen, sulfur, and oxygen are all going to be electronegative. So that means it's going to act very similar. They can pull electrons towards the electronegative. And then you have R group. Okay, so these are the things that are going to be similar and different between all amino acids. So when we look at example, here we have cysteine. So the first part is pretty much all the same. So you look at here, this is always going to be the same for all amino acids. Here's the main carbon that we're going to look at. So when we do drawings, this is always going to be in the middle. You're going to have a carboxyl group on one end, COOH, and then a amine group on another end. Normally when we write this, it's going to be more like this. You're going to NH2C COOH and then R group. So a lot of times they'll write it like that. This one's kind of on its side, but this is kind of has a better connotation because all the R groups are facing down. So you might see it one way or the other, but it's all very similar. Here's another example. So instead of amine group right here, a sulfide group right here, we're going to have an aromatic, which we're going to talk about. But you'll notice that everything else is the same. You have a amine group and a carboxyl group. Amine group and a carboxyl group. So all amino acids have the same thing. They have a carbon in the middle, a amine group and a carboxyl group, and then the R, which is what makes it different. So when we're talking about the 20 different amino acids, this is what's going to be different right here. What we talked about Friday is that we have something called chiral or non-chiral. So these are chiral because when you look back, you have on this alpha carbon right here, this one right here, you have 1, 2, 3, 4 different things on it. Hydrogen, our carboxyl group, amine group, and a R group. The R group can be the other ones, but most of the amino acids have a different R group. So because of that, we have a chiral amino acids. Most living things are going to be in the L configuration. So if you get something in the D, most likely you cannot use it. So when we have food, all our food's going to be in the L. Here's a good summary of what we're going to talk about. I'm going to try to categorize all the amino acids as in nonpolar, carboxyl group, amine group, aromatic, or hydroxyl group. And then we have others. But most of them are going to fit in these functional groups that we're going to talk about. So here's the first one. Here's an aromatic. Aromatic just means that they have a ring. And the reason why it's called aromatic is if you ever smell any of these, they have a very fragrant, flowery kind of smell to it. Some are bad, but they will have that certain smell to it because of that aromatic. So aromatic just means most likely it's going to have a 6-ring carbon with double bonds in between, and that's an aromatic. So you'll see that. Most of the time this is very unreactive because of carbons, but then here you have an OH group, which means that it can be reactive. Notice on these pictures that you have a negative here and positive here, which is a little bit different from what we had before. I'm going to say most of the time we can ignore what's going on here. And the reason why we can ignore that is it's going to be bonded to other amino acids. So when we have amino acids, we really don't have one. We don't care about one. We usually have chains of thousands. So these two right here are going to form the chain of amino acids. So even if it becomes ionized, so if it's by itself, it does become ionized, but we don't really care about that. What we care about is the R group. So we want to know, what happens to the things that are hanging off. Okay, but these are all aromatics. These, it's called allophilic, which is technically just means non-aromatic, but these are our non-polar. Polar, I know I squished it together, I apologize. So that means that all these don't really have anything that's electronegative. So when we look at these boxes, so once again, we're going to ignore the base of the amino acid. We're going to look at just the R group, which is highlighted in green. You'll notice nonpolar, 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 nonpolar. This one kind of, but since it's sandwiched between carbons, not going to be as polar. We're only going to see it more with hydrogen. Here we have nonpolar. So these are technically your nonpolar ones that are around. So they really don't do much. All right, here we have, let me get rid of this. We have carboxyl groups right here. Because we have carboxyl groups, when we have it in our blood pH, so that's what it means right here. If we have in our blood pH, it's going to actually change its charges, and that's what's going to be important here. So when it says physiological pH, that means pH of our bodies. Within the pH of our bodies, what's going to happen is because oxygen is so electrically negative and this one's positive, what's going to happen is that it's going to give away its hydrogen because it doesn't mind. It wants that extra electrons anyway. So what's going to happen is in this picture, you have a very negative oxygen. So you have 1, 2, whoops. 1, 2, 3, 4, 5, 6. And what makes it even more stable is this could kind of jump around, which we'll talk about resonance later. But oxygen's totally happy with having that many electrons. Hydroxyl group. Oops, let me get rid of this. So you have a hydroxyl group right here, which is an OH. So when you have an OH, it's going to be, here's tyrosine again, even though it was an aromatic, you're going to have a partial positive, partial negative, and so it can create hydrogen bonds. So that's the only thing you want to notice. Whenever you see imbalance of sharing of electrons, it's going to either be polar, which means it'll dissolve in water. It could create hydrogen bonds, or it could be acidic. Okay, so even though these don't gain or lose a proton or electrons, it is still able to do hydrogen bonds. Here's the sulfide group. It's very similar. It could be ionized, or it could create a hydrogen bond. Here we have carboxylamine, which just means you have a carboxyl group with the amine group. And you'll notice that it's going to have similar effects. You're going to be able to create hydrogen bonds with it. Here we have amines as well, and without the carboxyl group. And that one itself is going to be ionizable. So with amines, it's going to be the opposite of oxygen. Instead of gaining electrons, it's willing to gain hydrogen. So these are technically bases. So that's it. What I'd like you to do is kind of go back to this slide and kind of just label all your things correctly. So I asked you to make a chart the other day, so I'd like you to go through it, label them as either non-polar, carboxyl, amine, aromatic, hydroxyl group, and just kind of talk about or label it like that, so then you could put non-polar or polar. All right.